Maraba virus progresses the cure of HIV-1 by Rosanna Demircan. HIV is an RNA virus with two types, HIV-1 and HIV-2. Since HIV is a single-stranded virus, it indicates as having a positive ssRNA polarity and is in the Retroviridae family of the lentivirus genus, which can cause infection and be contagious to others. Symptoms include fever, chills, rashes, night sweats, muscle aches, sore throat, fatigue, and swollen lymph nodes as displayed in the picture to the right. This disease is able to worsen by resulting in AIDS, when HIV begins upsetting the host's immune system, causing terrible bacterial, viral, and fungal infections, which may lead to death. HIV can be spread through sexual contact, pregnant ladies infecting their offspring, and if it is injected into the bloodstream by a contaminated needle. Cancer can develop if the virus continues to progress, because as it weakens the immune system, the body is not capable of defending itself. The picture below shows how HIV can worsen and become AIDS when the proper medication isn't taken. The figure on the top left shows the HIV virus structure. Many treatments are in progress to find a cure for HIV, including the kill approach that uses a virus to treat another virus, which in this case, the Maraba virus or MG1 is used. It is considered to be a negative sense RNA genome, meaning that they have single-stranded genomes that help messenger RNA stay synthesized. This virus is a vesiculovirus, so it is usually found in mammalian hosts such as cattle and rodents, resulting in ulcers or oral vesicles. Oncolytic viruses or OVs, such as this one, can infect, replicate, and kill malignant cells. While doing this, they are capable of leaving normal cells unaffected. This is the reason why numerous OVs have been used and become progressive in advanced stages of clinical evaluation. The two pictures on the right show the structure of the Maraba virus, and the one on the bottom left shows how the OVs help the healthy gene and damage the ones that carry diseases. Nishchal Ranganath and his biological team created an experimental research trial in Ottawa Hospital in order to try and use Maraba virus to cure HIV-1. The goal of this experiment is to determine whether MG1 is able to minimize or fully get rid of the bad cells, U1 and OM10.1, in subjects with HIV-1 while keeping the good cells, U937 and HL60, intact. The infected subjects were sustained and separated to find out which ones were healthy or not, and the activated ones were injected with the HIV stalks. The experiment was done in vitro, which means that the testing was done in an artificial environment where the HIV-1 cells were extracted from the bodies instead of the tests being done on the bodies themselves. The cells were then passed for up to 18 hours in order to enter the exponential growth stage before the MG1 or mock version of it was added to them. They were left for up to 28 hours before being washed by PBS and plated for 2 hours before MG1 infection. They were left for 48 hours after the infection, washed twice by PBS, and stored at negative 80 degrees Celsius in order to help determine integrated HIV-1 DNA and viral outgrowth assay. All this was done by ELISA, an enzyme-linked immunosorbent assay used to target and determine soluble substances like antigens. Gel electrophoresis was used to keep track of the changed RNA and DNA in the genome. The figure shows the statistical evidence of the positive progress that MG1 infection caused by damaging the disease-containing cells while helping the healthy cells. The Maraba virus is one of the antiretroviral viruses that have made an impact in the death tolls of individuals who are diagnosed with HIV-1. According to UNAIDS, the survival rates of HIV-1 are increasing because it was able to kill around 1 million people in 2017, which is about a 50% decrease compared to the death rates it had on individuals in 2005. This shows that fighting a virus with another virus is possible, with the goal of decreasing the harmful cells without harming the healthy primary cells. The rhabdovirus MG1 has been successful at doing so, in order to target faulty cell communication and result in less of the spread of harmful viruses caused by HIV-1. In conclusion, the purpose of this experiment was to cure or lessen the amount of harmful cells, U1 and OM10.1 cells, in HIV-1, while keeping the good ones, U937 and HL60 cells, by using another virus, the Maraba virus. The kill approach was being used to reverse viral latency and aid immune-mediated destruction caused by the HIV-1 infected cells. MG1 has proven successful, which is why it is being used to experiment further on metastatic solid tumors and non-small cell lung cancer. The discoveries that the MG1 has made open doors to more MG1 therapy, along with the new findings it has taught biologists and scientists. This study shows how hope that the Maraba virus and many viruses may not only help people with HIV-1, but it may be helpful in curing other diseases as well. The figure displays a decrease in death rates of HIV-1 patients from the years 2000 to 2017. These are the references I used in my PowerPoint. Thank you for listening to my presentation. I hope you enjoyed it.